Today, we're going to present a study of the Purinosome that explores two articles. The first one being reversible compartmentalization of de novo purine biosynthetic complexes in living cells, and the second article being transiently transfected purine biosynthetics enzymes on stress bodies. So the picture here is a purinosome or a multi-enzyme complex that carries out de novo purine biosynthesis in the cell. And more specifically in the cytoplasm, and past studies have explored the function that each of these individual proteins, including the pathway, do, but little evidence is given to support that the proteins work together as a complex or together as a purinosome. And so this pathway here illustrates the 10 steps of the de novo purine biosynthesis, and the studies here will focus on the specific enzymes H-Driftguard and HFGAMs. And so for the first article, the hypothesis, it claims that the association and dissociation of the purinosome is influenced by varying purine levels in the environment.
So in order to support the formation of the purinosome, the enzymes previously mentioned were tagged with green fluorescent protein and orange fluorescent protein. Additionally, the HeLa cells were grown in either purine-rich or purine-depleted media to assess the influence of the varying purine conditions. After tagging those proteins, they were transiently transfected into cultured HeLa cells, and that process can be seen in image A. The transient transfection method protects the enzymes from degrading or leaving the pathway. Then in image B, we see the mechanics of a fluorescent microscope, and image C demonstrates the overall principle of fluorescence. So fluorescent recovery after photobleaching, or FRAP, is used in conjunction with confocal microscopy or Z-scanning, and both of these methods are used as a means of analyzing the data. So the confocal microscopy works by rapidly scanning the samples, collecting the fluorescence, passing through the pinhole, and then recording the image in the detector. Then FRAP is utilized to avoid photobleaching or damaging of the fluorophore. And that happens through selecting a region of interest and then bleaching it with an intense beam. Then the recovery is measured over time.
So for the data analysis and results, A and C in figure 1 show GFP-tagged enzymes of interest in purine-rich conditions, while B and D show the same enzymes in purine-depleted conditions. Both enzymes show an increase in clusters in purine-depleted media as compared to purine-rich media, which was expected from the hypotheses. And G shows H-Driftguard, and H shows HFGAMs, and I shows the merge image of both, and this confirms that the clustering, and also shows that there's an association of the enzymes within the same area, which is, in other words, colocalization. And so for the remaining 4 enzymes in the biosynthetic purine pathway, they were then investigated under purine-depleted media by tagging them with green fluorescent protein, as shown in the right column, and they were all co-transfected with HF-GAMs with orange fluorescent protein, which was shown in the middle column, and the merged images are shown on the right column. And this data suggests that these 4 remaining enzymes also co-clustered with HFGAMs under purine depletion.
So although that previous study supported that the punctate body formation was due to purine starvation, This second study speculates that the purinosome is formed from other factors such as non-native protein expression, stress, or aggregation. So this image just visualizes the 6 enzymes that make up the purinosome in the cytoplasm. All right, so an immunofluorescence assay was performed on the HeLa cells to determine if markers of aggregates, such as chaperone proteins and ubiquitin, co-localized with enzymes of the purinosome. So the leftmost columns show the enzymes tagged with green fluorescent protein or red fluorescent protein. The middle columns show the aggregation markers tagged with the GFP or RFP. And then the last columns will show red and green if no co-localization occurs, while an orange-yellow color is displayed if there is co-localization. So in figure 1, the 3 enzymes were all co-localized and co-transfected with the markers of aggregation. And in the second figure, the GFP tagged enzyme was co-localized as well with the chaperone and ubiquitin antibodies. The bottom row of the second figure were controls and assayed for non-markers of aggregation, so therefore they show no co-localization. So if purinosomes are formed by aggregation, it's possible that it is stress-induced, and therefore we would expect the presence of chaperones and ubiquitin proteins. The condition of aggregation was observed by treating cells with hydrogen peroxide to induce stress, and the results found that these treated cells showed aggregation regardless of varying purine levels. So therefore, results of this study suggest that formation of purinosomes, despite purine depleted conditions or the presence of chaperones or association with ubiquitin, indicates that purinosome formation is a result of overexpression and form in order to be degraded.
So the conclusions of each research article are as follows. The first article concluded that the purinosome formation was regulated by the stimulation of the de novo purine cells, and all 6 enzymes involved in the purine biosynthetic pathway co-clustered with HF GAMs in the purine depleted media. And the second article concluded that the punctuated bodies actually form independent from the purine presence in the environment due to induced stress. And this was done by adding the presence of the chaperone and ubiquitin. And cells with fewer purinosomes had showed a greater survivability than those with less, with greater number.
Some limitations to the study include that the purinosome may also just be a general phenomenon in all cell types during specific stages of the cell cycle, and also other non-explored factors might be involved in the regulation of the purinosome, such as phosphorylation of the enzymes. Lastly, while it is possible that there are endogenous purinosomes, the existence of purinosomes might be explained just as protein aggregation. And here are our citations.